Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, today in this lecture we are going to talk about a secondary growth in dicot root. In dicot stem, secondary growth, types of wood, that all we have discussed. But here today we are going to discuss the secondary growth or girth-wise growth or thickness growth or growth in a thickness or girth we can say in case of the dicot root. So if we talk about this, in the dicot root, the vascular cambium is completely secondary in origin. Dear student, in dicot stem, we have discussed that it is partially secondary in origin. But in dicot root, it is completely secondary in origin because it arises later in life. From the starting of life, there is no presence of this secondary vascular cambium. It originates from the tissue located just below the phloem tissue or phloem bundles, whatever the tissue which are located just below the phloem. We know that in root there is a radial arrangement of vascular bundle where xylem and phloem are present at the different radius. So wherever the phloem patches are present, below that you will see, or inside to that you will see the presence of this vascular bundle. This particularly tissues, and that tissues are going to form. Now these tissues are known as conjunctive tissue, which are present below the phloem. A portion of pericycle tissue is present, which is present above the protoxylem, outside the protoxylem, that form a complete and that particular also differentiate and start dividing. Now, by the fusion of this conjunctive tissue and the cells of pericycle, it form a wavy ring, and this wavy ring is going to become a circular. So, further events are similar to those already discussed above in the dicot stem. So what are the differences that we need to look down? Secondary growth also occur in stem and root of gymnosperm. In general, secondary growth does not occur in monocot. Okay, so this is the epidermis, this is the cortex, this is cambium ring, wavy cambium ring, which is formed here. Okay, and this is the endodermis. This is pericycle, and this is a protoxylem. So it is difficult to understand here. So let's understand it. This. So in dicot root, first we are going to talk about the formation of vascular cambium ring. Now, when we talk about the formation of vascular cambium ring, we have just studied that this vascular ring is completely secondary in origin, and this is formed by fusion of cells of conjunctive tissue and cells of pericycle. So what happens if we see the vascular bundle in case of the root? You will see such type of arrangement, that is radial, and in radial you will see here this phloem, and here you will see the presence of xylem. So this is the phloem, or we can say primary phloem, and this is primary xylem. Now here, if we talk, the cells of conjunctive tissue are located just below the primary phloem. So the cells of the conjunctive tissue differentiate, and here on the outer side you will see the presence of pericycle. Now, if we talk about this pericycle, the cells of pericycle which is present outside the protoxylem, because in root the exact condition is there. So protoxylem is on the outer side. So outer the protoxylem, this is the cells. This cells de-differentiate and form this cambium. And due to this wavy cambium ring is formed. Okay. So out of this, so first, what is happening? First of all, the tissue located just below the phloem bundle, that is a conjunctive tissue, become meristematic in the dicot root, and they form a separate curved strips of vascular cambium below the phloem bundle. Then, after the cells of pericycle lying above the protoxylem become meristematic and form additional strip of cambium, they fuse and form a wavy ring. The portion of vascular cambium that is formed by pericycle is less, but the main portion of the vascular cambium is formed by the conjunctive tissue. So this wavy cambium ring is in beginning, but later on it become circular. So later on it become circular. Okay. So this is the formation. Now when we talk about activity of this vascular cambium in dicot root, activity of vascular cambium in dicot root. An activity of vascular cambium in dicot root, if we talk, it is more towards pith. It is similar to the dicot stem, so more towards pith. So secondary xylem formation is more. On outer side of vascular cambium, it form secondary phloem. It form secondary phloem. So the activity of vascular cambium is root, similar to the vascular cambium of stem. Secondary xylem is obviously formed towards the inner side. Secondary phloem is towards the outer side of vascular cambium. The portion of vascular cambium which is formed by a pericycle is responsible for formation of the pith rays. Now, one more important point: portion of vascular cambium portion of vascular cambium formed by pericycle is responsible for formation of pith rays or medullary rays. And this particularly pith rays or medullary rays are made up of parenchyma. And these pith rays are known as primary medullary rays. Okay. Okay. This pith rays, these are known as primary medullary rays. Okay, whereas remaining vascular cambium that is formed by the conjunctive tissue form secondary medullary rays. So two types of medullary rays are found in secondary structure of root. And these two types of medullary rays are found in a secondary growth in dicot root. And this is the basic characteristic of root. Whereas in dicot stem we have studied that only secondary medullary rays are formed in the dicot stem during the secondary growth. And this both primary and secondary medullary rays, if we talk, conduct water and food in radial direction. Both medullary rays conduct water and food in radial direction. Okay, so this is the formation of vascular cambium and its activity. Now we are going to talk about the formation and activity of cork cambium in dicot root. Formation and activity of cork cambium or phallogen. Dear student, if we talk about formation and activity of cork cambium, then here in root, dicot root, cork cambium develop from pericycle. Cork cambium develop from pericycle. And this cork cambium, if we talk, it form cork on outer side and secondary cortex on inner side. Okay, so this is cork cambium, cork and cortex all are going to form a peridot. But dear students, when we talk about lenticels are found in root, but less in number. Okay, and if we talk about the cortex, because they are developing from the pericycle, okay, and pericycle is inner to the endodermis and cortex is present outer to the endodermis. So cortex, which is the outer side, which completely degenerate in root after the secondary growth of 1 or 2 years, and it fall down due to the pressure of the cork which is developing on the inner side. Okay, so this is the how the particularly cork cambium is arising. So dear student, here cork cambium arising from the pericycle. So this is the pericycle is intrastellar. Whereas in dicot stem we have studied that cork cambium or phallogen arise from the dedifferentiation of the which one, cortex cell, outer cortex cell or the hypodermis. This secondary growth, if we talk, this secondary growth is essential in root. Secondary growth is essential in roots to provide strength to provide strength to the growing aerial parts to the growing aerial parts of the plant and fulfill the requirement of water and mineral. Okay, dear students. So this is the thing. So it is important to the which provide strength to the growing aerial parts as well as it needed for the requirement of completion of the requirement of water and mineral. Now in dicot stem we have studied about the annual rings, but in dicot root, dear students, it is one of the important thing that generally clear annual rings are not seen in roots, are not seen in roots and are not affected and are not affected by changes of environment. Dear students, so this is what about the particularly dicot root. In dicot root, all cambia, either it is a vascular cambium or core cambium or pith rays or medullary rays, everything is secondary in origin. And dear students, this is all about the what secondary growth in the dicot root. In monocot, secondary growth does not take place. Okay, so that is one of the important thing that you should remember. Okay, and that is all about the what we can say the secondary growth in dicot root. Thank you.